We have some time for questions now. One of them is, what do dengue fever, West Nile virus, hepatitis C, and yellow fever have in common that makes them all part of this drug search?
All of those viruses are part of the same viral family, and their genes are fairly similar. The way their genes are laid out are pretty much the same, and the viral replication process is the same. All of are very similar, I should say, and all of them rely on that protease for their viral replication. So it becomes a very good shared target between all the different all the different viruses in the same family. In fact, the family name is Flaviviridae, which refers to the yellow fever virus that was the first one discovered in that family.
Okay, and what drugs are used today to treat these diseases?
Um, for yellow fever and West Nile and dengue, there really isn't any drugs. As I noted earlier, there's nothing that can be done. Every once in a while, ribavirin will be given to people, but generally that's not very effective treatment at all. And that's a general antiviral that's used in specific cases, but pretty much there's nothing, and that's why we're doing our work to find it. Now with hepatitis C, there's been a lot more work, and as far as I know, there aren't any protease inhibitors on the market, but they're very, very close to getting hepatitis C protease inhibitors on the market.
What are the symptoms that people get in more detail when they get dengue fever?
I'll first say that I'm a computational guy, not a virologist, so I can answer that. From what I understand from the people I've talked to who've had it and my research on it, it's like a very, very, very bad flu. Um, so I mean, it's running kind of the runny nose, general kind of stuff. The real key difference, the thing that's very prominent is, and why it's called breakbone fever, is just the skeletal muscle ache that you receive. It really puts you out. You can't move much, and it runs about a 2-week course, so it's very, very miserable of a disease, and of course if it takes you out, that has a strong economic impact, especially in developing nations when workers, if you have an outbreak, workers just can't do anything. So yeah, that's it's from what I've heard. I've had a friend who picked it up when he was in South America. It's pretty miserable, but it's very, very flu-like.
And someone was asking if there's something you can do to further improve the database of information you're building.
I have to ask in what way would that are they interested in? I mean, what we are doing is continually with the different types of the different levels of simulation and computations. What we're trying to do is enrich the database for compounds that will potentially bind, and so we're actually looking at continuing other methods. I won't share with all that right now, but we keep thinking about better and better ways of trying to do the drug discovery approach. We've been encouraged by what we've seen with the free energy calculations, but of course everything can be improved.
Somebody would like to know how long it will take to get to an actual drug. Can you describe the process after the project to
Yeah,
getting to that point where we can actually treat the disease?
I can get you some ideas of kind of what that whole process is like. First thing, it takes a very long time. It's about 10 to 15 years, I think, is the current pipeline. If we get a compound here, what we find a hit, and we found a few from some virtual screening that we've done. What we do is we spend some time characterizing it, and then when we test, we actually usually test both in vitro, which means we make sure that it inhibits the enzyme, and then we go and test it in cell culture assays to make sure that it works there. And then we have to do animal testing to see if it will knock the virus down in animals. And if those all pass, what happens is you kind of go through a drug discovery development project where you will take that existing compound and work on improving it so that it would be, you can hopefully give it orally. It would be ideal that it has a good safety profile. After that, you have to do a lot of extensive testing. Then you can apply for a application with the FDA, at which point then you go through the entire FDA approval process, which is generally done in 3 stages. Phase 1 of your clinical trials, you give it to healthy adults to make sure it won't make them sick if they're healthy. Stage 2, you give it to people who actually have the disease. And ideally what we imagine a drug to be like would be is that, as people start developing the symptoms, they are able to take it, and it mitigates the progress of the disease. Or if they're in a very severe stage, that we can knock what's the viremia down, which is the amount of virus in the blood. Phase 2 trials, like I was saying, they work on, you do it against people who actually have the disease. In phase 3, you do it with people with the disease, but it's a much broader study. And then after that, it has to go. Then it gets final approval, and it has to be continually monitored. So, drug discovery is actually quite the process of going through, and quite an intensive process. And really, our goal here, and what we're working with, is to up the chances of find as many possible compounds in as much chemical diversity as possible, so that we can increase the likelihood that we can get a drug all the way through that process.
Okay, and we have another question. What is the success rate of virtual screening versus traditional drug design so far in the medical industry?
I'm trying to think if there's a good study on that. Virtual screening has been successful, and the most most well-known cases are with the HIV protease drugs. Actually, been quite successful in the virtual methods used for discovering those. Most drugs actually come through a process of kind of everything. We're doing virtual screenings as well as doing biological assays simultaneously to try to identify and build the best drug possible. So both methods actually are pretty good. I'd say that for a virtual screening project, if I go through the literature and look at all the virtual screening projects and how many biological compounds they find, it's usually just a handful for screening several thousand compounds. So as we up our compound database, that increases the probability of helping us, or increases the probability of having good binders in the compound database, but also makes the challenge of finding them better and being harder.
One of the members is commenting that most of the areas of the world that are affected by this disease are economically challenged. And so he was asking what kind of things you can do to make sure, as a researcher, that some of these cures get to these disadvantaged places.
As a researcher, I think the thing that we can do best is really identify compounds that have the highest potential of becoming drugs. Because there are pharmaceutical companies who work on like dengue. Novartis had a project on that, but economically, it's not necessarily in their best interests. And I mean, they're corporations. They have shareholders, so it's kind of an interesting challenge for them. It's one reason why we, as an academic organization, decided to tackle this. And so, what we need to be able to do is find compounds that are as much a sure thing as possible. And so we work hard actually on figuring ways to screen that, and the academic community does to find the best compounds possible. Because the lower the cost to the corporation to develop, the more likely we would be able to actually develop it. And of course, there are been philanthropic efforts to push this kind of work forward.
Another question is, how long would this have taken to do without the virtual screening? I mean, somebody would have tried to do something, and how much money was saved by doing the virtual screening?
So, I've actually done the math. That's going to be quite a bit of money. If we have to do the methods that we use, our experiments, it's not terribly expensive. Though buying compounds can get real expensive to buy the compounds. We could have only screened maybe a few hundred compounds altogether, and we've been able to already look at and go through several million compounds and already reject a whole bunch of those. I mean, we still have to do experimental testing, and we have. I mean, we've tested about 50 compounds to date so far. We're still hoping to find and it's a field that's plagued by false positives, but as we've avoided having to test millions of compounds. So it can save quite a bit of money to do the virtual screenings up front, and allows us to really explore lots of chemical space that we would otherwise not be able to explore easily.
And somebody was asking if any of the compounds that you've screened have gone into assays yet.
Yes, we've tested about 50 compounds so far. A lot of those compounds were from phase 1, and we don't have a high expectation rate of those ones working just because just the natural issues of enrichment. We're looking really forward to getting the things that we've enriched from phase 2 and testing those, and we're in the process of actually doing that now. And so, yeah, we have tested compounds, and we've been successful with previous virtual screens too, in identifying potential inhibitors for dengue and West Nile. And we're in the process of developing and advancing those compounds.
And how much longer do you think it'll take to finish the virtual screening we're doing on World Community Grid?
I think we got a few more years to go through a lot of the database and do these free energy calculations. A lot of these free energy calculations in scale. But we're getting to the point now where we have lists of compounds we want to test, and so we're really moving into a very active testing phase here in our laboratory.
Oh, can you finally contrast what you're doing compared to using vaccines?
Yeah, vaccine development is something I'm definitely not an expert in, and it's a very complicated process as well. The difference is that a vaccine is a prophylactic treatment. You can give it ahead of time to prevent the spread, and ideally, what would be best, of course, would be a vaccine. The challenge is that for dengue especially, there's 4 different serotypes or strains of dengue that make that very hard. You have to give immunity to all of them. You can't just give 1 or 2. It actually can possibly make the viral infections worse. And there is some progress in that realm of doing the vaccines. An antiviral, on the other hand, is a lot more designed to just treat people that have the disease and have the outbreak to mitigate the symptoms of that disease. So you can think about it with the most commonly known drugs are the antivirals, of course, the HIV antivirals. And because we don't have a vaccine for that yet, we can't prevent people from getting HIV. But if they do contract it, we have treatments that can help mitigate the progress of that disease.
Okay, well, thanks again, Robert, and all those attending. Sorry if we didn't get to your questions. You can post additional questions on our forums at worldcommunitygrid.org, and Robert and the other scientists can answer those there. So this concludes our webcast, and thank you very much for all attending and contributing to this project.